Right, we're going to now take a look at one of the most important neuroimaging techniques. These are means of visualizing the nervous system from two perspectives. First of all, anatomically, in order to find out what the bits and parts are, and then physiologically, as we try to observe the brain in action, as it were. The two technologies we're going to be dealing with now are very, very similar. The first is anatomical, and it's magnetic resonance imaging, or MRI. The second we will get to is physiological, and it's functional magnetic resonance imaging. MRI, or magnetic resonance imaging, is done in a scanner. Scanner is a device routinely used in medical diagnostics. Many of you will have been in one. We use them to image all kinds of parts of the body, not just the brain. In order to conduct a scan, the patient or client lies on the central beer and is inserted into that borehole, surrounded by this large device inside which there are magnets that rotate at high velocity, making a frightful noise. So it's a bit claustrophobic, and you must remove all metal before going in or it'll be torn out of your body. So it's not a very pleasant place to be, but it gives us very, very high quality images, and it does no damage to the body. This is not X-rays, so there's some confusion here. X-rays are an much older technology used to produce a certain kind of image. X-rays damage the tissue that they pass through. MRI does not. It uses, and I'm just going to say, it uses complicated physics in order to produce these images. Now, we could go into the detail here, and I would be bluffing because I frankly don't understand the complicated physics. Most people using this technology don't understand the complicated physics that is used to produce the images, which is a source of some concern. Basically, what it's doing is using a very, very high intensity magnetic field to alter the spin of hydrogen ions or protons. The magnetic field used is vastly higher than the Earth's magnetic field. And from the interaction between the ions and the magnetic field, an image is produced. How voodoo is that? Which provides us with a great deal of detail, right down to sub-millimeter level detail. So we can image the places in the brain extremely accurately. It's slow. It takes about 15 minutes to get a good structural image of the brain. And these are what they look like. So here's a transverse cross-section through the brain. You can see the eyes there towards the top of the picture, and the nose. And you can see the ventricles in the middle of the brain. Here's another cross-section taken differently. This is a sagittal cross-section through the middle of the brain. You can see the nasal cavities. You can see the brain stem, the cerebellum tucked underneath the brain. And there's one other cross-section that's possible, of course. We've seen the transverse, we've seen the sagittal, and now we're going to look at the coronal cross-section. This is a coronal cross-section which you can pass through the brain like this. And if you download that brain tutor I mentioned in a previous video, you can pass through the brain along any of these three axes. So we can get very, very high quality structural images in order to find out what's there. And that's very important because people's brains differ. Your brain is quite different from my brain. The major structures are going to be shared, but when you get down to the level of detail, there's nothing shared between brains, really. You can't associate a single nerve cell in my brain with a single nerve cell in your brain. So we need to map individual brains in order to find out who we're dealing with. But these are all anatomical images. They don't show us anything about the brain in action, as it were, its functioning or its activity. For that, we need a different approach, which is called functional magnetic resonance imaging. This is also conducted using the same kind of scanner, and it uses the same basic principles as magnetic resonance imaging, but it employs a different signal. Specifically, where MRI uses hydrogen ions as the basis for generating an image, functional magnetic resonance imaging uses the oxygen level of the blood. So it is a signal which is dependent on blood flow. It's called the blood oxygen level dependent signal, or BOLD signal. And the idea here is that we might create experimental conditions, contrasting experimental conditions, and look at how blood flow differs in one condition compared to the other. That's not a straightforward way of looking at brain activity, and the images that we obtain are difficult to interpret. The way this works is we first obtain a structural MRI. So if we have someone who we wish to use as a subject in an experiment, we spend 15 minutes getting a good overview of their brain so we know what their brain looks like. Then we produce experimental conditions with at least one contrast, and we look at regional blood flow in the two conditions to see where the blood flow differs. It takes about 15 seconds to get one scan, so it's much faster and cruder than the MRI scan. So we've good spatial resolution, but very, very poor temporal resolution. So we could get one scan over the 15 seconds, but anything that's faster than that, we don't see. So we start, as I said, with an MRI, and then we have contrasting conditions, and what we plot over the MRI is shown here in color, and these are areas of the brain in which there is more regional blood flow in one condition compared to another condition. That's what these images mean. They do not show the brain lighting up. They don't actually register nervous activity at all. They don't register any neural firing. What they show is in one experimental condition there was more regional blood flow than in another. These kinds of images inflame the imagination, and journalists deal badly with them. They are often misreported in the press. They are very difficult to interpret, and we need to be very, very careful of avoiding the phrenological mistake of associating a place in the brain with a function which we have projected onto the brain. This image shows you two-dimensional slices to the brain. The data obtained is three-dimensional, so we can have wonderful resolution. We can see distributed activity throughout the brain. It's a wonderful technology, and it's continually getting better. You should know the basic structure, which is first to do an MRI scan and then to contrast two experimental conditions. We typically need a lot of repetition, so we might do 150 low-res scans, 75 in one condition, 75 in another condition, and then we compare the regional blood flow across the two conditions. The smallest unit in a volumetric scan is called a voxel, which is like a pixel on a two-dimensional image. A voxel is a three-dimensional cube, and we can get down to, again, about a millimeter or sub-millimeter, cubic millimeter average. Here's a study image from a study in which there was two conditions. In one of the conditions, someone was looking at a black screen. In another one, they were looking at a complex moving stimulus. And what you're seeing there, the orange area, is areas of increased regional blood flow towards the occipital lobe in the brain, which is unsurprising if you're watching something. So, we need a lot of caveats. A great deal of averaging in time is necessary. So the two experimental conditions require a lot of repetition, which means you can capture nothing of the ongoing creative sense-making activity of the nervous system. Rather, you capture only these reproducible effects. Brains differ greatly in their fine structure, so averaging across subjects is very, very difficult. Blood flow is very difficult to interpret. We do not know the relationship between the activity of neurons and the associated blood flow. That's an area of current research. It's highly controversial. Our ignorance here should be a word of caution that we are not reading brain activity. And we need, of course, to avoid the mistake of the phrenologists, which this kind of work sometimes encourages, which is the premature association of function with location.